Need a haircut.
Okay, so mitosis is a reproduction or division of the nucleus. When you start, you have one nucleus. When it's over, you have two. Now it's important to remember that once you have two nuclei, the normal procedure is cytokinesis. Cyto means cell, kinesis means division. So when cytokinesis is over, how many cells do you have?
Two.
Two.
What's the definition of psychokinesis?
Somebody raise a hand.
What did you say, psycho?
Cytokinesis.
What was it?
I hear you better when you know.
What was it again?
Division of the cytoplasm, division of the cell. There are some cells which have more than one nucleus. Didn't know that, did you? That's why I want you to make sure you understand. Mitosis almost always is followed by cytokinesis, but not always, most of the time. There are several different phases to mitosis. I'm going to go through them fairly quickly. Four that I want you to know are prophase, metaphase, anaphase, and telophase. Prophase, metaphase, anaphase, and telophase. And this is not a cell bio class, so we're going to hit this pretty quickly, okay?
Can you repeat those one more time, please?
Prophase, metaphase, anaphase, and telophase, T-E-L-O. All right, during prophase, the chromosomes condense. The chromosomes become visible. You can use whatever term you want to, put it in your own words. But the whole idea, if you're looking at a cell, you're not going to see the chromosomes unless it's in mitosis. Prophase is where they become dense and visible. Metaphase, they line up in the middle. They line up in the middle.
You said prophase lines up in the middle?
Nope, metaphase. During anaphase, they pull apart. They pull apart, kind of like this. They're lined up. Metaphase is easiest to identify because they're just lined up dead center. Then in anaphase, they start to pull apart. In telophase, they decondense. They diffuse back out.
What did you say? Say that again. Say that again, please.
In telophase, they decondense. They diffuse back out. Don't worry about getting too much detail on those. The main thing is chromosomes are visible, chromosomes line up, chromosomes pull apart, chromosomes no longer visible. My cell bio prof is probably shrieking in his grave hearing me go through a decimal. But we don't need to know it any more detailed than that. Is everybody okay with that? Does that help you, Chloe? Is everybody with me? We're not so much concerned with the details. I can always tell if somebody is a cell biologist or a geneticist by this process. How much do they get interested in it? Geneticists really focus on this whole process right here, mitosis. Cell biologists look at the full cell cycle. The easiest way to understand this is to draw it. So I'm going to do just that for you. Put a circle here. And we'll start right there about the 10 o'clock position. New cell, fresh, ready to go. The very first period of time is going to be the G1 phase. Growth 1 or gap 1. I don't care which you put, G1 is all we really need to know. This is basically normal cell function. The cell is doing what the cell does. Everybody clear on that? Thumbs up. If it's a bone cell, it's being a bone. If it's a blood cell, it's being blood. You get the idea. However, if the cell intends to divide, it's going to enter what we call the S phase. The S phase. S stands for synthesis. Now, as you know, synthesis means you're making something, right? What you're making is DNA. This is where the DNA replicates or doubles. The DNA replicates or doubles. Now, I want to point out something. Humans have how many complete sets of chromosomes? 2. Very good. May surprise you to realize you are not a simple one set of instructions. You actually have not one, but 2 sets, complete sets, on how to be a human. That's pretty cool, isn't it? You get one from your mom, you get one from your dad. And it's the interaction between those 2 that makes you, you. The environment always plays a role, the genes always play a role. Everybody clear on that? So how many sets of chromosomes do you have after S phase? Well, if normal is 2 and we double that, you now have 4. Does everyone see? And the reason is, since we're going to divide into 2 new cells, we need to double it to make sure the new cells each have 2 full sets. Everybody cool with that? Now, S phase is just the doubling of the DNA. You can put doubles the DNA, replicates the DNA, synthesizes more DNA. I don't care what you put as long as you understand that's what it is. Everybody clear on that? Generally, the transition from G1 to S means the cell is going to divide. Some cells never get to the S phase. We say those stay in the G0 phase. Instead of calling it G1, we call it G0 because it never intends to divide, and that's a good thing. The function of an erythrocyte, a red blood cell, is to carry oxygen. You don't want those red blood cells dividing in your bloodstream, do you? You want them doing their job, which is to carry oxygen. So we would say that the erythrocyte stays in G0. Does everybody see the difference? It may seem like we're arguing semantics here, and to a degree we are, but language is important. Using proper terms is important. That's why I'm having you all study those little vocabulary lists at the beginning of each chapter, right? You're building your knowledge base. Now, the next phase is going to be called G2. G2. This is the final preparations for division. We have all the proteins we need. Check. Are we big enough? Check. Is the DNA doubled? Check. You see where I'm going through all these. Do we need another cell? Check. It's like a checklist and a final chance to get everything ready to go. After that, you go into M phase, which is just another word for mitosis. Cytokinesis happens alongside mitosis. Almost immediately thereafter, how many cells do you have? 2. You started with 1, you've now got 2, the cycle begins again. Everybody see that? Now in humans, there's this thing called stem cells. Stem cells. Stem cells are pretty amazing because they can potentially become any cell type. You need another erythrocyte? No problem. Boom. You need a leukocyte, a white blood cell? No problem. Boom. You need an osteocyte, a bone cell? Boom. No problem. You see where I'm going with this, right? Stem cells basically represent potential. Potential. Without getting into the genetics of it, imagine that there are a series of switches. If we throw the right switch, we go this way. If we throw the right switch, we go that way. If we don't throw either, we continue on. You see how this goes? Once you throw a switch, though, you're locked into that path. You don't get to go back. Is everyone clear on that? This process of specialization, in other words, setting the switches, is called cellular differentiation. Cellular differentiation. Would y'all like a real life example that'll freak you out, make you smile? Okay, now I want everybody to participate in this. So if I don't see a hand going up, I'm going to get on to people. How many of you in this room begin your existence as a female embryo? Trick question, right? Even the guys have put their hands up. Did you know that? The default setting for a human is female. There is a specific gene called TDF that is located on the Y chromosome. Now, y'all know that I'm from Oneida. That's just south of here. You know the railroad is just over the hill, right? It runs fairly close to my house. It's the main line between Cincinnati and Chattanooga. So if we wanted to go to my house, we could all just hitch a ride on the railroad, couldn't we? Y'all know how railroads work. They have switches, don't they? Well, let's say we get down about Whitley City or Stearns, and somebody throws the switch from south to east. Instead of going south to Oneida, we're going to end up taking a different track and going east to Williamsburg. Does everyone see that? That's not where we want to go. It doesn't matter. We're locked on to that path. TDF is like that switch. Once it is thrown, it is an irreversible process. And now you know why men have nipples. Would y'all like me to explain that? The development of breast tissue begins before the switch is thrown. Once the switch gets thrown, it stops further development of that. Everybody see how that goes? But the potential remains there. There's a condition called gynecomastia, which is development of breasts in men. Did you know men can get breast cancer? It's not nearly as common as in women for obvious reasons, but it is possible. So everybody understand how this goes? I want you to understand that everything ultimately is under the control of the genetics. But there's a lot that goes on in development, isn't there? There are things that can interfere with that developmental pathway. Anybody here ever heard of Swyer syndrome? Swyer syndrome is where the individual has genotype XY, but phenotypically, meaning in terms of appearance, they develop as female. The reason being, something interferes with the expression of TDF. Della-Chapelle syndrome is the reverse of that. You can have someone who is genotype XX and they phenotypically develop as males. Generally what happens is you get that small chunk of the Y chromosome as the TDF gene relocates to an X and gets passed on. So there's a lot of variation in this pathway. There's a lot of regulation, and make sure you underline that word, regulation of it. Because do you want your cells to continue going through this process and dividing? Everybody wants to be young forever, don't we? Like Keith Richards says, nobody wants to get old, but nobody wants to die young either. Well, the reason we don't want our cells to continually divide is because otherwise we continue growing. Does anybody in here know who Andre the Giant was? You ever seen the movie Princess Bride? He was a big guy. He had a condition where he had I think it was a tumor on his pituitary gland. So it interfered with the regulation of the growth hormone. So he continually grew. Now, it gave him certain physical advantages compared to other people, yes. But it also meant he died much younger. I think he was in his 40s or 50s when he died. The world's tallest man ever. He was 8 feet, 11 something odd inches. He died when he was 22. You don't want to grow too big, do you? The other thing, and this is a biggie, cancer. Cancer happens when the regulatory mechanisms of the cell cycle break down. Make no mistake, you want a certain amount of cell division, right? You all are still growing, aren't you? Your brain doesn't fully develop until your mid-20s typically. Do you realize that? My niece is 10. I kid that kid, and she's growing like a weed. It seems like every time I see her, she's growing another inch, and I'm not joking. I used to carry her around on my shoulders. Now she's about here. She keeps going, I'm gonna be looking in her eye. It's a good thing. We want to grow, but eventually we want to shut it off. You only want replacement. Does everyone see that? Make sure you get that down, make sure you understand it.
Can you say again the definition for cancer?
Uncontrolled or unregulated cell division. For example, bone. I don't need any more bone in my body, do I? I'm fully grown, I'm 55, I'm fairly tall, I have as much bone as I want or need. We all agree on that? So we don't want my body making more osteocytes, right? Now let's say I fall and unfortunately I break my arm. I want to turn that path back on, don't I? Because I want to produce some cells to fill that gap, heal that break, right? But then I want it turned off. That is normal human function. When that gene gets turned on and left on, you get cancer. Somebody here likes to drive fast. Nobody would admit to it. I see a couple of it, yeah. We went south and hit that straightaway towards Footleigh City. There's a couple of nice open spots. Speed limit 55. I won't lie. When I hit those spots, I'm probably not going 55. It's probably up in the 60s. Get home a little quicker, nobody complains, right? If I had a couple of you in the vehicle with me, I doubt you'd be freaking out. If I were doing 120 through that stretch, you might not be so calm, right? Because it gives me less time to react. It means if we do leave the road, have a wreck, it's going to be more dangerous. You see where I'm going with this? Imagine if I put you into the seat of the car and I pushed the accelerator to the floor and locked it there. It's going to be a long, hard ride, isn't it? Like all the traffic through town. You think you could make it without wreck? That's what cancer is. Everybody clear on that? Now, cancer typically has 4 stages. Stage 1 is just abnormal cell division. Abnormal cell division. At stage 1, it's not causing any problems. Let's say you got a mole on your skin. You notice that mole's getting a little bit bigger, it's getting a little bit more discolored at the margin. Something just doesn't seem right. That's stage 1 cancer. You go get it check, doctor does a biopsy, cuts it all, the whole thing out, says, yeah, it's cancerous, but no problem, we got it all. Do you have to worry about that when you're my age? Because it's gone. See what? Stage 2, it's now definitely cancerous. It might begin to form what we call a tumor, T-U-M-O-R.
What was stage 2?
2.
What was stage 2?
Tumorigenesis, formation of a tumor. Now again, we're painting with a pretty broad brush here. We're just talking about generalities. This is not a cancer class. But I want y'all to at least have an understanding of what it is, okay? At stage 2, that little mole that was getting darker and had an irregular margin is now a fairly large bump that you know was not there a year ago. And sometimes it cracks and bleeds. Sometimes it hurts. It's definitely something's going on there. Everybody see where I'm going with that? It's a problem. Stage 3, let's just call it invasion. In stage 3, that tumor is now moving into the space occupied by a different tissue. I'll start getting weakness in my hand because that tumor has started effectively pushing my muscle aside, saying, hey, I need more room, get out of my way. You see where I'm going with this, don't you? Might even begin growing into the muscle. And it's important to understand it's still dermal tissue, isn't it? Because it started as skin. It's just going into the muscle. Stage 4 is called metastasis. Metastasis. When you hear a tumor has metastasized, you have reached stage 4. At this point, the tumor has fragmented. Chunks of little bits and pieces of it have broken off and they've been carried throughout the body to other locales. And once they get lodged in that other locale, they're going to continue growing, aren't they? So what started as a tumor in the skin of my right forearm has now become a tumor in my left kidney, my liver, my brain. There is no stage 5 because after that, you're dead. Now, again, this is a very, very large generalization here. There are no start and stop signs in this process. It's a continuum, right? You might have it detected right at the borderline of stage 1, stage 2. It's a judgment call on the pathologist to state which stage it's in. I just want everybody to go, it doesn't go stage 1, stop, stage 2, stop, stage 3, stop. It's a continuum. Is everyone clear on that? Treatment. Let's see, number 1 preferred method of treatment for just about any type of cancer.
Chemotherapy,
Nope,
radiation,
nope,
surgery.
surgery. Cut it out, it's gone. And this is a bit of a trick question. That's why I want y'all thinking about it. If it's stage 1 or even stage 2, it's really not that bad if you get it cut out. Again, very generalized because Getting something cut out of your arm is a lot different than somebody going fishing and cutting out part of your pancreas. But the whole point here is you remove the cancer and some of the surrounding healthy tissue to ensure that you have completely removed it. Now, the big thing, once it is gone, it is gone. Can you see that scar on my forearm? Some of you can see it. When I was 19 or 20, I think it was first fall semester of my junior year, I had noticed that I had a bump that hadn't been there before. It wasn't a big deal. I just rubbed my head over it and I could feel something in there. Went to the doctor over when he said, and he said, yep, there's a bump there. Gee, thanks, doc. He said, well, to know what it is, we need to do a biopsy. In other words, cut it, take a little piece of it, stick it in the microscope. And he said, it's so small, when I do the biopsy, I can actually take the whole thing out. I said, yeah, that sounds good. So he numbed me up local, stretched it out. I actually watched him do the surgery. We talked about it. Tiny thing, wasn't it? Stage 1 too. Do I have cancer today? I think about it once or twice a year when I'm teaching this part of anatomy. Otherwise, I don't even think about it. I've had it there 30 years. I'm used to it. It's no big deal. Everybody see where I'm going with this. So stage 1 or 2, generally you're able to get it with surgery. Almost always they'll try to do surgery first. Now, you mentioned 2 other things. Chemotherapy. Does anybody know what that is? If there was ever anything that was misnamed, it was chemotherapy. Chemical therapy. What it amounts to is you are taking poison. You're taking so much poison so quickly and in such doses that you are systematically killing cells within your body. Think of the typical side effects of chemo. Hair loss is a very common one, right? The reason for that, your hair follicles are some of the fastest dividing cells in your body. Gee, it's almost like those drugs that you're taking are targeting the fast dividing cells, right? Does that sound like cancer? That's why a lot of people have chemo lose their hair. Another very common side effect is nausea, vomiting, diarrhea, digestive system malfunction. That's because the epithelial cells that line your digestive system, guess what? They're also fast dividing. You see where I'm going with this?
A lot of like nosebleeds and stuff, right?
You'll have to speak up here.
A lot of nosebleeds and stuff. 'Cause
A lot of
your nose is
nosebleeds.
very dry.
You have to replace your red blood cells. You see where? Good job. I'm not trying to frighten you here, but I want people to understand. Chemotherapy is literally just hooking yourself up to an IV full of poison. And you're hoping that you kill all the cancer cells and that you kill them fast enough that you don't kill too many of the good cells in the process. Make no mistake, the chemo drugs are a lot better today than they were years ago. They're targeted, they have a lot more efficacy, they're able to use smaller doses, less side effects. Whether or not chemo gets prescribed, very often it will depend on the type of cancer, where it originated, and what stage it's at. Everybody see that? The advantage of chemo, it targets the whole body. Radiation. radiation is generally used only for certain specialized cases. For example, a brain tumor. If you have a brain tumor that's buried fairly deep in your brain, they're probably not going to crack your skull open and go fishing. Brain surgery is not something you want to be just doing on a whim, is it? What they can do, they can focus radiation, literally use magnets and specific wavelengths and all that, to focus in on that specific spot and say kill all of the tissue within maybe a half centimeter of that point. It's pretty cool, isn't it? If it is, say, a thyroid tumor, the thyroid preferentially absorbs iodine. So then they give you a dose of radioactive iodine so that your body absorbs the iodine. It goes to your thyroid and it kills off cancer in the thyroid and over the thyroid. See where I'm going with that? Specialized. If it's stage 3 or 4, they may use a combination. It's not that big.
I wasn't looking horrified, I was just thinking.
Well, again, some of the stuff I talk about here will make you uncomfortable. That's okay, it should. But it's not to scare you, it's not to horrify you. If any of it ever bothers anybody, can you get up and walk out for a few minutes? Absolutely not. We've all had different kinds of trauma, we've all had different experiences, there's certain things we can't deal with, and that's okay. But I want everybody to at least have an understanding of what's going on here. If you have stage 3 or stage 4, it's almost always going to involve some kind of chemo. What is the best way to deal with cancer? Huh? Close. You want to minimize your risk, obviously.
Just like plant on your family, things like that, while you have time.
Understanding familiar risk, that's good. Well, let's see if we can rephrase this. Who's most at risk for cancer in this room? There's one person who has a greater risk of cancer than all of the rest of us. Who is it? Want me to point the finger out? I have the greatest risk of cancer. Why?
You're older.
I'm older. Age is the number 1 risk. I have been on this mud ball about 2.5 times as long as you all have. I have had about 2.5 as times as many opportunities to be exposed to things that could cause cancer. Does everybody see how that works? And that's assuming all things being equal. But you can't minimize the risk of age, can you? Unless you stop aging. But like I said, nobody wants to die young. Chloe?
What about genetics? Can genetics?
Genetics definitely play a role, especially in certain types of cancer. I've got 2 cousins, both of whom had breast cancer. Their mother had breast cancer, 2 of their aunts had breast cancer. Not surprisingly, they did some tests and got the BRCA1 mutation, BRCA1, which is very common. There's definite familial link there. They have an adopted daughter and a biological son. Their son doesn't have to worry about it. Their daughter, does she have to worry about it? She's adopted. So she does not have that marker. Whenever they're doing an assessment risk for cancer, they're gonna ask you a lot of questions. And essentially what it comes down to are exposure and risk. Exposure and risk. If I smoke 3 packs of cigarettes a day, am I gonna get lung cancer?
Possibly.
Possibly.
If you do it, you're.
I won't. You spoke up first, I'm sorry.
I just said it was more likely.
It's more likely.
If you're doing it like years upon years of like smoking many packs a day, you're more likely.
I would certainly increase my risk with it. Does that mean to you that I get it? I could still live to be 100 and never have lung cancer. But if you understand anything about probability, you know that I'm increasing the odds. Would everyone agree with that? So there's definitely an element of chance here. I want to make sure everybody understands that. It's just like wearing a seatbelt. I haven't had a wreck in 25 plus years. 2 years ago, almost to the day, I was going home from work, somebody pulled right up front of me, bam. I had to call my mom for a ride, my uncle brought her up. When they saw the truck, my mom burst into tears until she saw me standing on the side of the road, completely unhurt. I didn't even have bruises. Good thing I have a habit of buckling my belt right. Now, if I had not buckled my seatbelt 1,000 other times I've made that drive, it wouldn't have any effect with it. See where I'm going with this? It's only the one time that I needed it, unfortunately, had it on, between that and airbag outside. So that's why age is the #1 risk factor for cancer. You simply have more chances to get exposed to something that will cause it. This falls under what we call risk management. Risk management. When I was in the hospital with my staph infection in my spine, they did chest x-rays every 2 or 3 days. Every time that you have a chest x-ray, you are increasing the likelihood of developing some kind of cancer. Why were they taking that many? Because there's a far bigger risk of me developing pneumonia from laying flat on my back all the time. They judged the risk of cancer versus the risk of pneumonia and said it was worth doing. Everybody see how that goes? Had I not been so weak, had I not been in there for septic infection, they may not have done them so often, or they may not have done them at all. We all have different lifestyle choices that we make, don't we? Anybody here visit a tanning bed? Couple of you. You're exposing yourself to ultraviolet radiation. Does that increase your risk of skin cancer? But so, just walking on a beach on a sunny day without wearing sunscreen, you see where I'm going with this? And I appreciate everybody can share, I'm not making any judgments. Some of us judge that risk and we say that's acceptable, don't we? Because that's what we want, exposure to the sun, we want to get the tan, and that's how we look. That's fine, because you're making an informed decision. Everybody cool with that? So the #1 way to deal with cancer is early detection. Early detection. If you find it early, your options are much greater, your odds of survival are much greater, everything. Like I said, 25, 30 years ago, I've not even thought about it. You get it at stage 1, that's as good as it gets, isn't it? I've got a little scar, I've got a cool story to share with my students, no big deal. I know a person who ended up getting stage 2 skin cancer on the tip of her nose. By the time she bothered to get it checked out, they had to do so much surgery to clear the margins of the tumor that she ended up with a little, I hate to say crater, but that's basically what it was. They had to do a little bit of reconstruction. But it was still better than her losing her nose entirely tube and it's at the automatic being carried through her body. She's in her mid-80s today and survived quite well. I think again, what we're dealing with. Early detection is by far and away the best. It's one of the reasons why we tell you to make any pains you get, rashes, growths, things that just don't look right, don't feel right, there's probably a reason your body is in pain and telling you something. Okay, everybody cool, understand cancer? Next up, I want to talk about stem cells. The three different types we're going to worry about. First is called totipotent. Totipotent. Literally means total potential.
What was that word again? What was that again for the first one?
Totipotent. T-O-T-I-P-O-T-E-N-T. Totipotent. Literally means total potential. What can they become? Anything. They can become a skin cell, a blood cell, another stem cell. Heck, they can even become a person. Anybody here know any identical twins? Three of us. The rest of you don't know identical twins? They are the result of an embryo that split into two separate totipotent embryos, so they each developed into a person. Pretty cool, isn't it? When I say toady, I mean toady. Incidentally, if you want to have fun with somebody, ask them if they're for or against human cloning. Most people will say against, and just smile at them and say, what do you got against twins? Because identical twins are natural clones. Did you know that? Pretty cool. Totipotent stem cells generally means embryonic stem cells. From a research perspective or therapeutic perspective, they're the most desirable. But they can become anything. Now, remember though, those represent a clean slate. We've not flipped any of the switches yet, have we? Once you start flipping the switches, differentiation begins. Differentiation is an irreversible process. So most adult stem cells are pluripotent. P-L-U-R-I-P-O-T-E-N-T. Plural potential. A pluripotent stem cell can become any cell type or tissue type. What it can't do, or what it can't become, is a new person. Pluripotent stem cells have got a lot of therapeutic potential, but you also have to worry about the risk of cancer. Next up, you've got multipotent. Multiple potential. What does that mean? Well, it means they can become any cell type within a tissue. Any cell type within a specific tissue. An example would be something called a hematopoietic cell. And you don't have to write that down, I'm just giving you. A hematopoietic cell is a multipotent stem cell that's found in the blood marrow. It is the source of your red blood cells. and your white blood cells, and your platelets. Those are very different cells, aren't they? Red blood cells carry oxygen. White blood cells have an immune response. Everybody see where I'm going with this? Platelets are involved in clotting. The hematopoietic cells can give rise to any of those. Can it give me a new neuron or nerve cell? No. Because neural tissue is a different type. Does everybody see that? I need a pluripotent stem cell for that. Now, some people say that there is a thing called a unipotent or a monopotent. Uni or mono meaning one. And those are stem cells that can divide, but they can only become one thing. So does everybody feel comfortable with stem cells? Anybody have any questions over this? Okay. So let's review what we have learned today. First thing we talked about, let's just do a mini review right now. Is everybody okay? So you should know chapters 1, 2, and 3. So when you study this afternoon or tonight, make sure that you have no questions over those 3 chapters, okay? Make sure you've read all of them. Make sure you know the following bits of information from them. What is science? What is the scientific method? What is a hypothesis? What are the characteristics of a good hypothesis? What is polarity? Remember, water is a polar molecule, right? What does that mean? What does that mean?
What does what mean?
That water is a polar molecule. Well, it means that water has several properties that are important to life, right? It's got a high specific heat. It's a near universal solvent. It has surface tension. All those things we talked about water. Know your way around the periodic table. Make sure you have a copy of the periodic table to bring with you to the exam. A hard copy. I'll leave it your responsibility for that, okay? I'm not gonna hand them out. That's just something that you can get on your own. On that, you need to know the atomic number, the atomic mass, what an ion is, what an isotope is, what a valence electron is.
What kind of electron?
Valence VALENC. Know the three different types of chemical bonds from strongest to weakest. So that would be covalent, ionic, and hydrogen.
Strongest to weakest.
Strongest to weakest. Covalent are the strongest, ionic are the middle, hydrogen are the weakest.
I guess you repeat that one more time, please.
Covalent, ionic, and hydrogen.
And that was the three different types of,
Chemical bonds.
It was strongest to weakest.
Covalent, strongest covalent. Know how to determine the charge of an ion. Know how to balance chemical equations.
Know how to charge.
Determine the charge. If you missed that and you'd like me to help you with it, I will be happy to do so. Macromolecules. Number one, carbohydrates. Their function is short-term energy storage. To a lesser degree, structural.
Could you repeat that one?
Structural. Carbohydrates, short-term energy storage, structural. Number two are lipids, long-term energy storage, and insulation. Number three, proteins. Their primary function is catalytic. Secondary is structural. Everybody got that?
I didn't know what the beginning thing you said was.
Catalytic, did I not discuss that with you?
You did.
Enzymes. It lowers the energy of activation. It lowers the energy of activation. So I can't just cause that piece of paper to burst into flame, can I? I have to put energy into that by striking a match or using a lighter or something.
What did you say before, macromolecules?
I'm trying to remember, that's
Balance chemical equations.
chemical equations, okay. I've got a one-track brain. If you give me two stops down the road, I am lost. Okay. And then the last one would be nucleic acid. For DNA, the function is information storage. For RNA, it's information transmission. Amino acid transport. RNA is the molecule that does it all.
So the nucleic acid is DNA and RNA?
Know the three different types of transport into and out of a cell: passive, active, and semi-active.
So you said two inside a cell?
Into and
Into
outside.
and outside.
I'm going too fast. Know which one of those requires energy. Know the organelles of the cell.
Did you say passive active and proactive?
Passive, active, and semi-active. And then the stuff we talked about today. That help everybody? I will see you in lab. Question?